Hi, my name is Francisca Rodríguez-Cabello. I am a civil engineer in bioinformatics from Universidad de Talca, Chile. Today I will present analysis of differential expression in the phaseolin gene in samples of Chilean and foreign Phaseolus vulgaris L species. The plant material used in this research belongs to the Centro de Estudio de Alimentos Procesados. They correspond to 4 types of bean varieties of the species Phaseolus vulgaris L, each of them in 3 different stages of the maduration of the bean grain. The Coscoran and Tortola varieties are of the Chile breed that belong to the Andean gene pool. The Negro Argel variety of the Durango breed belonging to the Mesoamerican pool, while the Enriqueta variety is part of the Nueva Granada breed that belongs to the Andean gene pool. The Nueva Granada and Durango breeds in this research have been incorporated as a comparison with respect to the Chile breed. In the methodology, the reads of each sample were assigned to the reference genome with HiSat2. A file of annotated genes position was then provided, where the alignments were passed to a string type for transcript assembly. Later, with the assembled transcript to the abundance of this also in a string type was estimated, also providing additional read count data for each transcript required by Valgum. Finally, in Valgum, all the transcript and abundance of string type were taken, including the fragments per kilobase of transcript per million read sequence, FPKM, which were grouped by sample type corresponding to the bean varieties and their biological replicant number. Regarding the results, different expression analysis takes a white to red color from lowest to highest expression. In Figure 1, we see the analysis of differential expression with respect to the gene that encodes phaseolin in each of the samples used for each state in Coscoran bean. In Figure 2, we see in the analysis of differential expression with respect to the gene that encodes phaseolin in each of the samples used for each state in Enriqueta beans. In Figure 3, we see the analysis of differential expression with respect to the gene that encodes phaseolin in each of the samples used for each state in Tortola beans. In Figure 4, we see the analysis of differential expression with respect to the gene that encodes phaseolin in each of the samples used for each state in Negro Argel beans. In conclusion, this DAG analysis determined the genotypic difference with respect to the phaseolin between the 3 stages of the maduration for each type of bean. In the case of Coscoran bean, Enriqueta, and Tortola, the accumulation of the gene that encodes phaseolin decreases, while in Negro Argel the accumulation does not decrease. It remains constant, and analysis at more advanced grain state may be necessary to determine if it remains constant or decreases. The methodology used has made it possible to obtain the specific abundance of the study characteristics, making tables of characteristic expression per sample directly available. This facilitates the application of alternative differential expression tests or the evaluation of other statistical properties of the assembly in case they are required in subsequent analysis. Acknowledgement to Basilio Carrasco, Scientific Director, and Bárbara Arévalo, Research, both from the Centro de Estudio de Alimentos Procesados of the Universidad de Talca. I am attending to your questions. Thank you very much.